The broadcast is now starting. All attendees are in listen-only mode.
Hello everybody, good morning, good afternoon, good evening, depending on where you are in the world. I'm Beck Newton, and on behalf of Scientific Update, I'd like to welcome you to this webinar. So it's entitled Leveraging High and Medium Throughput Automation for Process Development at MSD, and it's sponsored by Technobis. So before we start, I'm joining from sunny Manchester here. We'll just get some admin. So we're recording this session, and this will be shared with you later, along with a PDF of the slides. That should come through via email. Sometimes it goes into spam, so just watch out for that. If you also can find it on our webinar archive area of the website, if you prefer to do it that way. If you've got any sort of technical problems with the webinar, then my colleague Claire is online in the background. So if you use the chat function, then she will hopefully be able to help you with that. Sometimes it may be a case of just logging off and on again and resolves any issues. Otherwise, questions for Maddie, if you could please type those into the question area of your dashboard. I will then pass on those questions at the end of the talk. So that's the admin side of things. So now it's my pleasure to introduce you to our speaker today, Maddie Deem. So Maddie earned her PhD from the university. She was working in Professor Jason Hines' laboratory. During her doctoral studies, she was making use of lab automation platforms to generate data-rich reaction profiles, which was then being used to help with mechanistic understanding. In 2023, Maddie joined the group at MSD, so also making use of data-rich experimentation. So yes, thanks again to Technobis for sponsoring this, and I'll now hand over to Maddie to give her presentation. Thanks, Maddie.
Okay, great. Yeah, thanks for the introduction. I'm really grateful for the opportunity to give this talk today. And I'm excited to share a little bit about how we implement automation at different stages of process development in the data-rich experimentation group at MSD. Before I jump into any chemistry, I just want to quickly describe our group and its mission. So the data-rich experimentation, or DRE group, is part of a larger enabling technologies organization, which works to develop new technologies and also deploy existing technologies to support our partners in kind of other parts of the drug development pipeline. We really work most closely with process chemistry and kind of process biology and chemical engineering, and we're involved with projects across all stages of process development, which calls for many different types of tools and technologies. The DRE group works with a wide range of technologies, which range from custom hardware and automation to data science and data engineering solutions. But the focus of my presentation today will just be on some of the lab automations in our group. To begin, I'll introduce a couple of the platforms that we use during different stages of process development, starting with early process development. So early process development work often requires a really large amount of chemical space to be searched and then narrowed in order to identify feasible routes or optimal conditions. And in many groups, this work relies heavily on small-scale, high-throughput experimentation, which goes hand in hand with automated liquid handlers. We favor Hamilton's, Tecan's, and ChemSpeed's, to name a few, in the DRE group. But really, the precision and the accuracy of these types of instruments while handling small volumes, coupled with the flexible and highly customizable deck layouts, make them ideal for designing custom screening workflows. Additionally, early process development work usually means that material is pretty limited, and so working on small, high-throughput scales can allow for more data to be generated from small amounts of materials. Then as the projects turns into or sort of mid-process development, reaction optimization becomes key, and we start to leverage more medium throughput tools like the AmicoChem or the ReactALL. And during this stage, it's important to begin to understand reaction robustness, which can mean looking at the reaction performance on larger scales or probing mixing sensitivities and starting to understand the effect of overhead stirring. It's also really useful to collect time course reaction profiles during this stage to start gaining some mechanistic insights into the system. However, there's often still material limitations, and so that can make it challenging to collect really high quality data on a meaningful scale or in a scale predictive manner. And so medium throughput automated reactors like the ReactALL are becoming really critical technologies for bridging the gap between small scale early development work with larger later scale work. And then finally, as projects move into later stage process development, we tend to rely really heavily on easy maxes and accompanying in situ or online PAT that kind of go with that. As the throughput decreases and the material demand increases in this later stage work, we really try to focus on maximizing the amount of data gained from each reaction rather than maximizing the number of reactions that we run. Today, I'm going to present 3 case studies which demonstrate common workflows that the data-rich experimentation group uses and also highlights some of the lab automation that I've introduced in the last couple of slides. The first case study that I'll present describes the development of a high-throughput time course screening platform, which was used to study antibody-drug conjugate reduction reactions during kind of earlier process development work for this program. Just so that everybody's on the same page, I'm really briefly going to walk through the steps to synthesize these antibody-drug conjugates. So starting with a MAB, or a monoclonal antibody, which consists of heavy and light chains held together by disulfide bonds. A reductant is added to the system. It's usually some kind of phosphine reductant, and TSEP is a common example. This cleaves the disulfide bond, exposing thiol groups. And then a payload is introduced to the system, and that usually consists of a drug linker portion and then a kind of cytotoxic warhead on the end. The thiol will react with the drug linker, covalently binding the payload to the different chains of the MAB and producing our antibody drug conjugate. These ADCs can then be analyzed in a large variety of ways in order to quantify the extent of the reduction and the conjugation reactions. So ADCs are becoming increasingly common in our pipeline, and we saw the need to develop a platform which could really accelerate reaction discovery and reaction optimization for our partners in process chemistry and process biology. Specifically, we had a partner group come to us that were interested in trying to characterize the reduction portion of this reaction, and so we were kind of inspired to develop a workflow. We wanted to develop an automated workflow which could screen the kinetics of that reduction step, and we knew that there were several key features to include in the platform, the first of which being compatibility with a 96 well plate format, just for optimal data density. One motivation for creating this workflow was responding to a partner group's request to determine conditions suitable for translation to a continuous flow process. And so incorporating an aspect of time course sampling and kinetic analysis would be really important for that. The workflow that we eventually arrived at is summarized at a high level on the bottom of the screen. So beginning with plate design and plate dispensing, we would then sample the reactions over time, analyze all the data by HPLC, and finally visualize the resulting trends. So it seems, of course, quite simple when listed here, but I want to take a little bit of time to show some of the workflow development because this represents a really typical automation project opportunity for the data-rich experimentation group. Our first step is frequently narrowing down which hardware we're going to use. And so for this, we wanted to select the appropriate automated platform to use for the time course sampling. Because we were working in a 96-well plate format, we narrowed down our choices to automated liquid handlers, which were really compatible with plate-based sampling. We first considered a chem speed, but found that the time that it took to sample a plate was too long compared to our desired sampling intervals. And also, these systems are solvent-backed, which poses a risk of solvent contamination during sampling. So we next looked at a Tecan Evo and found that it had a lot of benefits, including faster sampling times and the ability to use work lists. However, the system is also solvent-backed. So finally, we evaluated a Hamilton microlab prep, which was really reliable and it was fast. But unfortunately, there's no ability to sample from the plate in the back, which is where the agitation and the temperature control occurs. And so we decided to move forward with our Tecan used for time course sampling, despite the risk of backing solvent contamination. Then with the hardware set, we started working through some of the details of the other steps, starting with the plate design. Historically, our group has used a software called Library Studios to design custom screens. And essentially, you just input your desired reaction conditions, and then the software will generate a description of the stock solutions and amounts that you'd need to dispense into each well to achieve the desired design. My colleague, Sarah Moore, who worked with me on this project, wrote a Python script which converts the design, the kind of human readable design from Library Studios into a machine readable work list. And we decided to then sort of upload the machine readable work list to Hamilton in this case to do the dispensing of the stock solutions. We didn't need agitation or heating for the kind of initial reaction plate setup step, and so we decided to use the Hamilton here because it's air-backed rather than solvent-backed. So we then transferred the prepared reaction plate to the deck of our Tecan, which added the final initiating reagent, most commonly our phosphine reductant, TSEP in the case of a lot of our screens, and then the Tecan would pull reaction aliquots over the course of the reaction and dispense them into other 96-well plates on the deck of the liquid handler, which contained quench and diluent. We would then take the 96-well plates containing all of our reaction samples and analyze them by HPLC. And as this is a plate-based workflow, we were generating a pretty enormous amount of LC data. And so we knew that incorporating some data analysis and data management feature into this workflow would be really important for its success. So my same colleague, Sarah, created a database that ingests the processed HPLC data. And then she also developed a nice front-end web app, which allows the data to be graphed and visualized in a number of different ways. This has also become kind of our centralized hub for storing all of the data from these types of workflows. And that's been convenient for our stakeholders because they can come to one place and really easily compare the results from different conditions within a screen or even between different screens or different programs. Okay, so that's an overview of the platform development. And now I'll show some results from one screen that we ran. As I mentioned previously, we were collaborating with colleagues in biologics process research and development and flow chemistry in order to identify suitable conditions to run this reduction process in flow. And more specifically, they were interested to know the impact that pH and buffer identity had on reduction rate. So we found a commercially available kind of pH and buffer plate, which was great because we didn't have to pH adjust 96 different things. And also conveniently, the team had previously established the use of N-ethylmaleimide as a surrogate payload. And so that allowed us to avoid any handling of potent compounds. With the screen objectives in place, we then executed our workflow, starting with the design and the dispensing of the stock solutions. And then we ran the reactions and sampled them over time, analyzed all the data by RPLC, and finally processed all of the trends and then shared them with our external stakeholders. This figure here is, one visualization of the data that we collected. This shows the LC area percent of the fully reduced MAB over time, and the color coding is according to the pH of the reactions. So if we just consider the end point of these reactions, it would appear that low pHs, which are shown in purple, performed the best, they gave the highest conversion, and that our higher pHs, which are shown in yellow, performed worse as they gave lower conversions. However, if we then consider the full time course, we see kind of a different story. So the lower pHs have relatively much slower initial rates and only reach that high conversion after several hours. And so they would not be a good choice for translation to a continuous process. And then the higher pH conditions have significantly faster initial rates, but again, the highest pH conditions really don't have a suitable level of conversion. And so this data was used to identify the ideal pH range for the team, which was between about 7.5 to 8, and that gave suitably fast initial rates and also an acceptable amount of conversion at the end of the process. This platform has now been used for a couple different ADC programs at MSD, and we've also expanded it to modalities beyond MABs and antibodies. But I really wanted to highlight this work for the first case study because I think it's a good example of the technology development work and platform building that we do. And it's also a good example of using high throughput experimentation and automation in early process development work. So the next project that I'll talk about really demonstrates the use of existing technologies for route scouting and reaction understanding. The compound of interest, MK8189, for this case study was a drug candidate for several different neurological indications. And it had undergone previous route development, but due to some issues with process robustness during scale-up, the synthetic route was being reevaluated. The specific fragment that I was working on was this chirocyclopropane heterocycle shown here. And a lot of the proposed routes in order to access this fragment came from the allylic alcohol starting material that's shown here. The first synthetic route we evaluated was using Chris Ueda's cobalt-catalyzed asymmetric cyclopropanation chemistry. And this was a really attractive route given how cost-effective base metal catalysis can be. And also Pfizer has shown that this chemistry can be used successfully on scale. However, our initial screening efforts really only yielded low to kind of moderate EEs for our substrate. And that's in line with what the Ueda group had found in their publication as well for their heterocyclic substrates. Additionally, there were some concerns with the robustness and the safety of using superstoichiometric amounts of heterogeneous metal powders on large scales. So we also considered a kind of classical Simmons-Smith approach to forming the cyclopropane. But in order to get even moderate EEs, we were needing to use stoichiometric amounts of very expensive chiral auxiliaries. And so this was not an ideal route for us either. Then two of my colleagues who were working on this fragment as well, Haifeng and Jiao, envisioned a very clever combined approach that would use the best aspects of the Ueda route and combine it with the most favorable aspects of the Simmons-Smith chemistry. So they wanted to keep the inexpensive base metal catalyst and chiral ligand for stereo control, but then use a zinc carbenoid as the carbene source, which would result in a redox neutral cycle and avoid the use of any reductants. Very excitingly, some proof of concept reactions showed good reactivity and moderate EEs for our heterocyclic substrate. And so we realized that we would need to really dig deep into this transformation, and there was a lot of kind of unexplored chemical space that we would need to map. And so we turned to high throughput screening. We began by screening different ligands, and we started with kind of a general pre-plated ligand kit, which was designed, dispensed, and then stocked in the catalysis lab, which is part of the data-rich experimentation group. So based on the hits from the initial general ligand screens, we identified two best ligand classes. So OIP ligands were found to give very high yields and high conversions, but relatively low EEs. And this was the ligand class used in Ueda's work, so the trends that we observed aligned with their results. And then also pyridine bisoxazoline or PIBOX ligands we found gave a large range of conversions, but tended to have higher EEs. And so we followed up with several more focused screens looking at just many different types of OIP and PIBOX ligands, and eventually identified this ligand here as the best in terms of EE and yield. We then moved on to solvent screening, and this is a co-solvent screen using THF as the main solvent component. From this, we identified a very clear trend in which the reaction really favored polar co-solvents that are shown on the left side of the plate. And my colleague Xiao worked really closely with our NMR group in order to try to understand why propylene carbonate or polar co-solvents in general performed the best. And they found that there was a big stabilizing effect from these solvents on the zinc carbanoid reagent. And so eventually we determined that a combination of 2-methyl-THF and propylene carbonate was the best for both promoting catalysis and also stabilizing that zinc carbanoid. With the ligand and the solvent set, we then finished out our screening campaign by looking at different cobalt precursors, and we found several metal precatalysts gave suitably high conversion and EE. So altogether, our high throughput screening efforts gave us conditions which yielded 95% assay yield and 98% EE. And this was great. However, as we started to translate these results to larger vial scales, we were seeing kind of bigger and bigger discrepancies between the assay yields from the high throughput screening and the vial scale reactions. And so it became really crucial to understand the robustness of this reaction. And we identified the ReactALL as an ideal instrument at this point in time because material was still very limited. The allylic alcohol material was not in large supply for us at that time. With the medium throughput automated reactors, specifically the ReactALL, which we used, we were able to generate data-rich reaction profiles using only about 100 or 150 milligrams of material per run. So this is the reaction profile using the conditions identified from our high throughput screens. And we observed initially very quick kinetics. The reaction reached about 75% conversion in an hour. However, the reaction then stalled, eventually stalling out at about 85% assay yield and 96% EE. So this result was consistent with other vial scale runs that we had done, but obviously there was a relatively large gap between the high throughput results and these results here. One other benefit of using the ReactALL is the ability to monitor temperature over time. And so we observed a very significant exotherm upon addition of the zinc carbanoid reagent, which we used to initiate the reaction. So that, along with some other previous observations, which suggested that maybe lower temperatures might benefit the reaction, we conducted an experiment in which we cooled the reaction during the addition of carbanoid reagent, and then slowly warmed it to room temperature over time. This approach resulted in a 97% assay yield and successfully closed the gap between the screening scale and our vial scale reactions. And we attribute the improvement in assay yield to better control of the exotherm and therefore better stabilization of the carbanoid species. Typically, generating a reaction profile like this with time course sampling, overhead stirring, and kind of a finely controlled temperature ramp temperature profile would require using a reactor that requires gram amounts of material. But again, this reaction really only needed about 100 milligrams of the starting material, which showcases the power of these sorts of medium throughput reactors for this stage of process development. We also explored the kinetics of this transformation using the same instrument, and this was beneficial for the team for gaining some early mechanistic insights. I'm not going to get too much into the weeds of the kinetics, but we found that there was a 0th order dependence in the starting material and a positive order dependence in the cobalt, the ligand, and the zinc carbanoid reagent. So giving them some early insights into possible turnover limiting steps in the catalytic cycle. Additionally, the team was very interested to understand more about the catalyst behavior, and they really wanted to know if there was a dimeric resting state of the catalyst. So we collected reaction profiles at a number of different catalyst loadings, and then we calculated the initial rates for each of these reaction profiles and plotted the initial rate versus the catalyst loading. And then this initial rate versus catalyst loading plot can be fit with 2 different equations, one which indicates a first order in catalyst, and the other approach which indicates a fractional order of 1.4 in the catalyst. So while we weren't able to determine the exact numerical order in catalyst, we did conclusively provide data for the team that showed there's no dimeric resting state, which is really the question that they were trying to answer. Through a combination of high throughput screening using liquid handlers and reaction profiling using the ReactALL, we were able to identify optimal conditions and gain some crucial reaction insights without significant material consumption for this program. All right, the final case study will focus on our process development efforts for an oncology API. Obviously, the structure is slightly redacted, so I'm going to do my best to make this still an interesting chemistry talk, but you'll have to use your imagination to fill in some of the structures. So the particular reaction of interest was an acid-catalyzed transformation, which was forming, a key fragment for the synthesis of the API. And as in the previous case study, the team first started with high throughput screening in order to identify optimal reagents and loadings. So they conducted an acid screen, which indicated that higher pKa acids shown on the right hand of the screen really performed best for the reaction. And then they took their 2 best acid catalysts forward and screened it in kind of a custom design screen, looking at acid equivalence and also co-solvent identity and co-solvent volumes. So in addition to the bond forming transformation that occurs here, this process is also a reactive crystallization. And so the co-solvent identity and volumes are very important for performance. This screen indicated actually that the highest product LCAPs were obtained with low equivalence of acid and no co-solvent. But as this was a crystallization, the physical state of the reaction was really important, and this gave kind of an intractable, reaction mixture. And so to have a good physical state of the reaction, the team moved forward with using 1 equivalent of acid and also 2 volumes of co-solvent. And the conditions that they identified from these 2 screens served them very well throughout most of process development. However, some issues started to arise during large-scale pilot campaigns, and specifically a key impurity was starting to form in greater amounts. And this impurity was not rejected during crystallization, and so it was getting carried through to downstream steps. the team really needed to de-risk impurity formation, and that's when the DRE group got involved. And again, we identified the ReactALL in this case to be ideal for this program. We chose the ReactALL because of its throughput, so you could screen 5 conditions at once, and while we weren't material limited, it was still important to find an instrument with increased throughput in order to accelerate our data collection. So we began by assessing the effect of temperature on reaction performance and impurity formation. And these are the product versus time profiles for the reaction at different temperatures. And it is very obvious that temperature is not an acceptable control strategy for this impurity, as even decreasing the temperature 10 degrees from the center point really leads to significant decreases in conversion and way too much loss of yield for the product. So we moved on to evaluating the effect of acid equivalence on impurity formation. And we monitored the reactions using acid equivalence from 0.3 to 1.5. And then we can see that with lower equivalence of acid, the reaction is slightly slower. However, if you really squint at this graph, it also appears that the product LCAP is slightly higher for the lower equivalence of acid, which I'll talk about more in a minute. And then finally, if you look at the impurity formation profiles, we can see that having less acid present really promotes the formation of this key impurity. Additionally, I mentioned earlier that the acid equivalents impact the physical state of the reaction. And so having less amount of acid really leads to a lot of solid formation, and again, just gives kind of an intractable reaction mixture that would not be suitable to run on large scales. So based on this data, it seems very apparent you should just maximize the amount of acid. It suppresses the key impurity, and it gives, a mixable reaction. However, as I mentioned earlier, the product LCAP was lowest when you had more acid present. And that's because if we look at the impurity versus time profiles for all of the other impurities which form in these reactions, we can see that having more acid really promotes impurity formation for everything except our key impurity. And so we needed to find an acid loading that could balance suppressing the key impurity with product yield, essentially, and not forming too many of all these other impurities. We conducted a follow-up screen which looked at 0.75 to 1.3 equivalents and found that 1.1 equivalents of acid was the ideal center point. These handful of ReactALL experiments were used to inform decisions during what we would call like pre-characterization work, and they saved a lot of experimental work or kind of future experimental burden for the process chemists and the engineers. Additionally, the conditions that were identified in these experiments were used for the process at the manufacturing site, and the key impurity was successfully controlled below its specified limit. All right, to summarize, we employ a variety of lab automation technologies in the data-rich experimentation group, and we've really tried to curate a collection coverage for that project. Sometimes, as shown in the first case study, this requires the development of new workflows and utilizes automation that's really flexible and customizable. And other times, as highlighted in the second and the third examples, we rely on automation that's more immediately deployable in order to meet pipeline timelines and still produce robust data. Okay, to finish, I'd like to thank my many collaborators on the case studies that I discussed today and all of the members of my group. I also want to thank Technovus and Scientific Update for sponsoring and hosting this webinar today. I'm really grateful to have had the chance to talk. And of course, thanks to everybody online for joining, and I'd be happy to answer any questions.
Great, thank you so much, Maddie. Really great overview there of the capabilities that you have at MSD. So we'll get started with some questions then. So what would you say are the biggest challenges for using lab automation based on yours and colleagues' experience built up over the years?
Yeah, there are a number of challenges. I think that, there can be pretty high upfront costs to installation and consumables, and there's an abundance of options, so it can be difficult to choose the right instrument to make an investment in. Also, something to consider is like how it integrates both with other automation that already exists in the lab, but also how it might integrate into the workflow of the scientists who are going to use it. So kind of like a change management problem. We think a lot in our group about, how to ensure adoption of automation and workflows that we build. And so we don't want, things to be just siloed or contained to our group. And so again, I think, that's important to have maybe a variety of options, some that are super customizable that require like a lot of subject matter expertise, and then some that are more quickly deployable, and which can be used, without as much like training for an end user. But yeah, I think those are some of the biggest challenges.
Okay, great, thank you. So given that you do have a number of these different platforms and tools, how do you go about choosing which are the best for given projects?
Yeah, we always start with a discussion with our stakeholders. So, projects are usually informed by like stakeholder needs. We would have a sit down meeting, we'd figure out what the exact problem is that they need to solve. And then we start kind of trying to narrow down which hardware we're going to choose based on like how much material they have, what kind of analytics they're going to need. Is it HPLC compatible? Will they need to like do a lot of post-processing to samples like filtration, derivatization, etc.? Do they want some kind of spectroscopic analysis like IR? Does it need to be NMR? All of these things really influence what we choose. And then also, throughput and timelines, and yeah, trying to gauge if it will be more beneficial to the team to collect a large number of reactions and just look at endpoint analysis, or if they want really detailed reaction understanding and like data-rich information about a fewer number of reactions. And that kind of informs where we'll, land on the scale of like automated liquid handlers to like the easy max setup with a bunch of PAT kind of pointed at the same reaction.
Yeah, okay, great, thanks. So question on the analytics. So in the case study 2, you showed some concentration data measured on the ReactALL. How were those data measured? Was that HPLC with an internal standard or some other method?
Yes, exactly. Yeah, so the ReactALL samples into little LC vials, and then we would put those onto our instrument and measure that way. We also use an internal standard just to like improve sampling accuracy and kind of calibration, although usually the sampling accuracy is pretty good. The RSDs that we get off that instrument are around like 1% or less.
Okay,
but yeah,
great.
that's HPLC.
We've got a question around what extent the team employ DOE algorithmic approaches versus expert-guided experiment planning, please, if you can comment on that.
Yeah, that's a great question. I really didn't get into like that aspect of our group at all. But or like maybe slightly cheesy slogan is we're trying to maximize, well, I guess I won't go to cheesy slogan, but part of our workflow really incorporates like a reaction planning and data interpretation aspect, which are usually computer-based. So if we're working with a team, we try to strongly encourage the use of, some kind of like statistically or algorithmically guided reaction planning, unless the team has an extremely specific question, like I want to know what the reaction looks like under these 5 conditions, full stop. But if it's like, I want to, or, you know, they want to characterize like, and find the best conditions, then we would try to employ like maybe Bayesian optimization or something like that. Or if we're working, you know, more towards the later stages of process development, then we'd probably incorporate like a formal DOE design there.
Okay, great, thank you. We've got a question here about different types of equipment. So what is the advantage of using the ReactALL instrument compared to 4 vials set up in an EasyMax, for example?
Yeah, so if you run, I guess, like vial scale in an EasyMax insert, you could certainly get the temperature control. You wouldn't necessarily have overhead stirring unless you have like a really customized setup. And also, I guess you could stick an easy sampler probe in there. But it's, I would say, it's difficult to have overhead stirring and sampling and temperature control and vial scale. So to us, that's the benefit. When we're material limited or we're like throughput limited, then, you know, we would turn to this instrument. Also, I mean, yeah, if you wanted 5 reaction profiles from like vial scale and EasyMax, you'd need 3 EasyMaxes and 5 easy sampler probes to run those like concurrently. So
yeah, okay, thank you. Another question here relating to analysis. So when you were talking about analysing assay yield and EE, when that was being done in the 96 well plate, again, was that HPLC data? Is that all automated? And what's the kind of turnaround time for a 96 well plate from setup to final data collection? If you can comment on that, please.
Absolutely. The analytical like HPLC analysis and processing is the bottleneck for us, especially for that workflow where we were generating, you know, if you want to take 10 samples, that's 10 plates worth of data. So that was all HPLC. Again, we had an internal standard and we were doing assay yield based on the internal standard. Yeah, the analysis took several days to run the samples. And then, you know, the data processing takes a minute as well. Yeah, I guess we just try to optimize and like keep the LC method as short as possible. But in a lot of cases, for us, the analysis time does end up being our bottleneck.
Yeah, I think that's a common observation, isn't it, with this type of work? Okay, question around the heat map visualization that you showed. There's a question about which software, although I think you said it was maybe some custom built software that was developed in-house for that.
For the first case study, sorry?
The heat map visualization, which I think, yeah, was on the first case
Yeah,
study.
that was a custom solution built in-house. So again, as I mentioned earlier, some people in our group are, you know, more like data science focused, and so it's common that there be kind of somebody from DRE who's like doing the automation, the physical lab work, and then partnering with somebody who can build like a nice back-end data pipeline with like a web app for visualization. I think Sarah made that in like Streamlit is what the web app was made in, but yeah, that's a custom solution.
Yeah, okay, thank you. We've got a question here. MSD appears to have a specialized group for HTE and automation that supports multiple platforms, so ADC, small molecules, etc. Are there significant synergies between these technologies?
Yeah, I guess synergies in the sense of like the technologies and the modalities. I mean, we definitely, you know, we strive to create workflows and find like automation that can be applied to many different modalities. Generally, the project needs vary slightly, but we have had pretty good luck in like using the same automation for a lot of different like modalities or partner groups. I don't know if that answered the question.
Yeah, I mean, I guess from what you've described, it sounds as though you have a centralized group that various stakeholders can approach, and a lot of the workflows are going to be the same across the board regardless of the type of project. Yeah, thank you. Let's see, what else do we have here? A comment on data management tools, perhaps. Obviously, there's a lot of data generated here, so what kind of data management tools, how has that evolved over the years as more and more data is generated?
Yeah, that's definitely a challenge. We do have people in our group working to create data pipeline solutions, but I think that's sort of the next frontier we're trying to tackle, is integrating or connecting the data flow, from the HPLC directly back into the planning software, or connecting it directly into an electronic notebook, and reducing some of that data processing and data transfer burden. I don't know that we have a great universal solution for it yet. A lot of it tends to be project specific, like the example that was shown with the database, where that's somebody who is building a custom solution for one workflow. But the goal is to have something like that exist on a much larger scale, where all HPLC data is tagged and kind of imported to a centralized database, which is accessible for people across the company. But yeah, that is very much a work in progress, I would say.
Yeah, okay, thank you. And we've got a question here that's around crystallization, perhaps a general question, in which case I'm happy to invite the Technovis team on if necessary. But the question is asking whether or not the ReactALL can evaluate the problem of oiling out during anti-solvent crystallizations, so that then the user would be able to try and understand or troubleshoot a crystallization problem.
Yeah, I haven't used the ReactALL for crystallization work too much, but there is a webcam in each reactor, and so you could theoretically monitor the physical state and see the oiling out occur. But yeah, I'll definitely let Carmen or Rena chime in for a more detailed answer on that.
Yes, indeed, we do. This is Carmen. We did have seen this with several customers, and we even have an ongoing project now at Rowan University where we were looking at this. Yeah, the added value of having the cameras there allows you to see quite a lot what is in the vial, and if the attendees or the person that asks is a bit familiar with our crystalline instrument. So the same technology of the camera from the crystalline is also on or available also on the ReactALL. So with different magnifications, color cameras, and the software package is also available on the ReactALL that helps you to look at the particle shape, what do you have there, sizes, and so on. So definitely it is a possibility for the crystallization world also, or oiling out, to explore the ReactALL in that direction.
Great, thank you. So I think we have, oh no, sorry, a question has just come in. So it's around using ligands on a small scale. So what kind of dispensing equipment? I guess that sort of speaks to the limitations and challenges sometimes, depending on the practicalities of what's being dispensed, whether you're using solid or liquid handlers. Are there anything that you've come into, any problems that are sort of repeat problems with handling of certain types of reagents?
Yeah, so generally for the ligand screening kits, larger scale stock solutions are made, and then a liquid handler like a Tecan would be used to dispense the stock solutions into a bunch of plates, and then we would Genovac down those plates and evaporate the solvent, and they get stored as very small amounts of solids in the plates. Solubility can definitely be an issue, and slurry dispensing is kind of a pain. We have different solid dispensing units, but they don't frequently go low enough to be useful for the screening kits. And so we have ongoing work to try to optimize slurry dispensing on a couple of different liquid handlers. But yeah, that that can definitely be a challenge. Typically, though, we try to just dissolve everything up as a stock solution and then dispense out that to the plates and evaporate it down.
Yeah, okay, thank you. We've got a question here around time-sensitive reactions. If you've got a long sampling analysis time for HTE, any advice for how to approach time-sensitive reactions?
Yeah, that's a tricky one. So, if the LC method is like 30 minutes and you're trying to do high throughput screening, that's really rough. Coupled with the fact then if you have poor analytical stability, I mean, if you have long sampling time, a long method analysis time, and poor sample stability, I mean, I think, yeah, the throughput has to change, or like you've got to find some quench or like derivatization that improves the stability of your sample. I mean, I don't know if this is a great answer, but if you've got multiple LCs available and you're using an internal standard, sometimes I'll like try to squeeze onto somebody else's HPLC if I have a really long like method analysis time. But that is a tricky scenario. I don't have a great answer for how to like more efficiently get through method analysis if you've got a really long analysis.
Yeah. Okay, thank you. And then we've got another one here that again maybe better answered perhaps by the Technobis team. So a question on the ReactALL being compatible with Raman. What experience do you have, Maddie, with integration of PAT, or perhaps the Technobis team want to comment further?
Yeah, I have never used the integrated Raman probes, so I'll let Carmen take that one.
Yes, indeed. We do have a few systems out there. I think your neighbors in New Jersey with Raman. But I would not know more than that. I am trying to think of a case study now. I know that it is used with the ReactALL, the Raman. So the ReactALL is delivered with the lenses inside, and the customer comes with their own Raman. I would not know to say which Raman they have coupled to the ReactALL, but they are units out there with this PAT, let's say. From a PAT point of view, we do have since recently, I'm also looking at Irina of what we have released, pH control. We are preparing for feedback on pH to be released now in Q1. The temperature measurement and control is already in the ReactALL. I think this is it, right, Irina? Yeah.
Okay, great. Thanks, Carmen. So I think we'll probably wrap up. Thank you very much for all of the questions that we've had in from the audience. Thank you, of course, to Maddie for your excellent presentation and to Technobis for sponsoring this. We'll be in touch with the recording and the slides shortly after. But thanks very much for joining, everyone. Hope to see you at another one of these soon, and enjoy the rest of your day.